Hi guys, Dr. Douglas Gillard here once again. It's time for part 2 of our continuing series on human embryology. In this video we're going to we just started cleavage, so we'll just back up a tiny bit and talk a little kind of refresh on cleavage, then go into implantation, endometrial change, and the formation of the bilaminar disc. And we'll almost get through week 2, but not quite. All right, here we go. Okay, remember from the last video, we had a oocyte that a sperm had gotten all the way into the cell membrane or into the cytosol, and its pronuclei dissolved for the most part. It lost its tail and it melted with the oocyte's pronuclei, and both of these were 1N, had a diploid number of 1N, or they were haploid, meaning they only had half the number of chromosomes that our cell should normally have, like a cell from our finger or nose or liver. They have 46 chromosomes. These guys have 23 apiece, but they melted together and these chromosomes were mixing, and so we got a fertilized oocyte at that point with a chromosome, 46 chromosomes and a diploid number of 2N, right? Or you could say 23 pairs of chromosomes is the AKA for 46 chromosomes. At this point, because we're in still, we've went through interphase already. We have 92 chromatids, but only 46 chromosomes. Okay, that's where we were. And remember, this is everything I just said. The nucleus of the newly formed zygote then completed the cell cycle. So this thing is now, instead of individually going through mitosis and cytokinesis, together this acts, instead of two nuclei, this acts as one big nuclei, and the whole thing is going to continue the cell cycle. And once it gets through cytokinesis, we have two clones, two cells, where this thing will split right in half. Well, let's go right to a picture of it. This is what we have, and the cell cycle has been completed, and then we have our first creature. This is a little bitty tiny human, I guess, in a way you could say. It's only got 2 cells in it, though. We need millions and millions of cells, so this process is going to keep going and going and going. But at this point, this embryo is cleaved, or what we can call the zygote. The zygote will cleave in half, and once cleavage occurs, then you can say that the embryo is here. This is an embryo. There are some AKAs you should know for embryo, which makes things confusing. One author calls it a conceptus, and other authors call it that too. Moore actually calls it a conceptus, and that's one of the chiropractic board books, so watch out for that name. And you can always, if you in doubt, you can always call it a zygote because it's still a zygote. Okay, that's about where we left off, I think, after the embryo being born. Then each one of these cells, each one of these blastomeres is now going to go through the cell cycle again. The chromosomes there's the nuclei of each cell. These are called blastomeres. So it's going to go through the cell cycle, and then these are going to split in half. Okay, so cell cycle repeats itself. Each blastomere goes through the cell cycle. And the cell cycle, of course, you remember that from histology and I think physiology, and you probably had that in undergrad. I'm not going to dig into that too much. But interphase, the genetic material doubles, meiosis, cytokinesis, it splits. And now we have a 4-celled creature. That's that is still called a zygote. It's nothing special yet. We're going to get a special name again. There's just a recap of the cell cycle itself, where it starts here in interphase, which is by far the longest, G1, S, and G2, goes into mitosis, and then cytokinesis is where it splits into 2 daughter cells. Or in embryology, we don't call these daughter cells, we call them blastomeres. Okay. And there's just the different, if you blow up mitosis and look at it, there's a prophase, metaphase. Some authors split it into a pro-metaphase and metaphase, then anaphase, telophase, and then cytokinesis. Okay, another point to make that everything is not so simple. I mean, in reality, one of these would be smaller, one of these would be larger. One of them might start the cell cycle quicker than this one, so you don't always have this nice, beautiful splitting. So therefore, you could get some odd number embryos. Like here's a 9-celled embryo, and that's because this embryo doesn't behave like it does on paper. It kind of has its own pace and speed. Okay, next theory is, or next thing we need to talk about is compaction. Compaction is interesting. So normally, up until about the 8-cell stage or 8 blastomere stage, these blastomeres are pretty loosely arranged within the embryo. They're not tightly bound together. And by the way, the corona radiata is for a couple days. It still hangs around. Zona pellucida hangs around for a long time. We actually get into the uterine cavity to get rid of that. We've got a hatch out of that thing, as we'll see. So those are still around, but the cells are loose. However, after the 3rd round of cleavage, the blastomere the blastomeres pack very tightly together, and that's called compaction. In fact, they start to get smaller too. So the overall cell of the zygote, the overall size of the zygote does not change. You think it would get bigger at each cell division, but it doesn't, at least not yet. The blastomeres get smaller and more tightly compacted together. That's called compaction. Compaction microscopically, now we have tight junctions that's formed between the daughter cells or the blastomeres. So that helps hold them together. But the key point is compaction. Blastomeres pack together. The embryo does not grow in size. It's still the same size. Now, about day 3.5 to day 4, we have a 16 blastomered embryo. And that gets a special name when it has 16 daughter cells within it. The early embryologists thought it looked like a mulberry, so they named it the morula. Morula, which is Latin for mulberry. Now, there's a little controversy about when the morula comes to being. Some authors say 32 cell stage. Some say it's somewhere between 16 and 32. Langman, which is a board book, says 16, so let's go with 16. Moore says 16 cell as well, so we can go with that as well. Okay, morula is here. Okay, here's a real human morula right here. And it is, yeah, this is the same cell. This is the same size. This zygote is the same size as the 2-celled one. These have just gotten smaller and more compacted together. Because of what phenomenon? Compaction. And the genetics of the morula, each one of these blastomeres is just like a cell in your skin, the tip of your nose, your liver. That is a normal diploid cell. Has 46 chromosomes. Its diploid number is 2n. And these are continuing to go through the cell cycle. So pretty soon this one's going to split in half. And they're always splitting and growing. Okay, so they're still diploid, each blastomere, 46 chromosomes, or you could say, some authors say 23 pairs of chromosomes because you got one for mom and one for dad. So it's the same. Those are AKAs. 23 pairs is an AK for 46 chromosomes. If you want to get technical, of the 46 chromosomes, 44 of them are called autosomes, while 2 of them are called the sex chromosomes. Here's just an overall spot of where we are. Day 1 is when fertilization completed. Fertilization actually started already, but this is when the embryo was born after we got our first round of cleavage, and then we had 2 daughter-celled zygote, and that's called an embryo. Day 2, each one of those daughter cells split again, and now we have 2 daughter cells, or you could say blastomeres is kind of an AKA. Should really be saying blastomeres. Day 3, each one of these is usually split by then, so we have 8. Day 4, now we have a 16-cell blastomere, and now we give that a special name called the morula. Okay, everybody good with me? And then while all that is going on, day one, by day two, the fertilized oocyte is moving, right? Its job is to get down this fallopian tube and get into the cavity of the uterus, the uterine cavity. And so day two, it's here. Day three, it's here. Day four, it's here. And then between day 4 and 5, it actually gets out of a tight sphincter here and goes kaplunk, and it's in the uterus, as we'll see here in a second. Now, one thing, let's take a time out here and look what's going on. By the 4th day, we actually have genes working and creating gene product, and that's pretty amazing. So the very first thing that happens, so all these things, normally not much is happening inside the nuclei inside here, but by day 4, the nuclei of each one of these things, they start doing their thing. Nuclei undergo transcription, and then you make an RNA transcript, and that goes to a ribosome. You guys know this. That goes through translation, and you get yourself a gene product, which is usually a protein. And that's already happening by day 4. And in fact, we can already see there's already 2 cell populations that have arisen by day 4, and we haven't even gotten into the uterus yet. So the genes are working, right? The genes are already turned on. In fact, you have 2 cell populations, a central cell population, which is going to be called the intercell mass, and then you have one that stays very peripheral, kind of on the outside of the ball, and that's going to be called the trophoblast. And when cells do different things like that, that's called segregation. So how did this segregation occur? Well, we just said some genes got turned off, some got turned on, and there's some proteins. These have some protein products, and these don't have certain protein products. But this is the earliest example of embryonic gene expression at day 4. So that's pretty amazing. So let's recap. So we have a inner cell mass, which are the cells that stay inside. Watch out for these darn AKs. The authors are terrible. Langman is a Board of Chiropractic Examiners board book, so you got to know embryoblast as an AK for inner cell mass. And how do you study this stuff? Embryology is not easy. You just got to make some note cards, and or however you do it, make your lists, and you got to memorize these names. The inner cell mass ultimately will give rise to the embryo proper. That's all the embryonic tissue comes from this inner cell mass. A little bit of the extra embryonic tissue does as well. Then the cells that stayed on the outside are called trophoblasts. Again, Langman, who is a Board of Chiropractic Examiners book, he kind of calls it, Langman kind of, and more too, they kind of use words that some of the other books don't. But I definitely want to point these out to you. Langman calls it the outer cell mass, but nobody else really does. So trophoblast is the best word for this. Moore's, the other chiropractic board book, calls it trophoblast. So trophoblast, inner cell mass. Okay, trophoblast becomes not intraembryonic tissue, but extra embryonic tissue, like the placenta will arise from this layer. Are we good? Here's a cartoon again, just to recap that. Here's a morula. It has working genes turned on. The genes, and we'll look at exactly what genes here in a minute, but the genes are secreting products that make these cells different from the cells out here, which are not secreting certain products, secreting other products, as we'll see. So segregation has occurred. The inner cell mass is already starting to form, and then the outer cell mass, or the trophoblast, everybody calls it trophoblast. And yep, zona pellucida is still hanging in there, right? The corona radiata is gone by day 2, so this is still hanging in there. Okay, so now watch out, nerd alert, but we got to go into this a little bit. Why do they segregate? So the inner cell mass cells is pretty simple. In order to become an inner cell mass cell, you have to have genes turned on that make 2 gene products or proteins, OCT4 and NANOG. It's as simple as that. So the cells in the inside that become the inner cell mass, they secrete OCT4 and nanog. Here's the central dogma is turned on. Everybody knows probably in junior high these days what the central dogma is. That's where we have machinery that reads the DNA on the chromosomes. So genes get transcribed into messenger RNA, which leaves in eukaryotes. It leaves the nucleus and it finds a ribosome. Ribosomes will translate that single mRNA message into the protein that they're destined to make. And in this case, they're making OCT4 and nanog. Therefore, they're going to stay inside the cell. Trophoblast cells, on the other hand, they have two different gene products. They have a CDX2 and GATA3 are their products. These ones, in fact, stop the secretion of OCT4 and nanog. So these block nanog and OCT4 and don't let it be transcribed, and therefore these cells kind of float to the outside and become trophoblast cells. Okay, simple as that. All right, so about day 5, finally, our morula has made it all the way down the fallopian tubes, and it has just got out this tight, there's a little sphincter here that holds, that makes it a little tough for these cells to get out. And so that had to be loosened up. Progesterone, which is a hormone secreted by the corpus luteum, it does the job there. This thing is loosened up. So it drops out of the fallopian tube and into the martini glass. Not really a martini glass, right? This is into the inside of the body of the uterus. So this is called the uterine cavity. It's filled with a salt water type fluid. And now we got our morula, like an olive in a martini, floating around in here. And we'll see what that does. So now we're going to form, the morula is going to turn into a different thing called the blastocyst. So once the morula is in the martini glass inside the uterus, it soaks in this salt water. Actually, sodium potassium AT pumps are born, and it literally transports that fluid, and fluid begins to accumulate inside the morula itself. That fluid accumulation will actually start to form a cavity. It pushes the inner cell mass cells over to one of the sides. There's the inner cell. I could have colored those. Those are inner cell mass cells. So the cavity is called the blastocyst cavity or the blastocele. Everybody good with that? It doesn't affect the trophoblast much, but now we have our first cavity. With the passage of time, that's going to become our primary yolk sac. But for right now, it's a blastocyst cavity, aka blastocele. You got to know both of these AKs because you don't know what board question it's going to throw out there, but they're pretty similar. This morula is now no more. Once a morula develops a cavity inside it changes names to the blastocyst. No more morula, blastocyst is here. Everybody good with that? Okay, next one. Here's a beautiful picture. There's a human blastocyst. We can see that the blastocyst cavity is nicely formed here. We have trophoblast around the outside. And there's our inner cell mass pushed up to this region right here. Actually, we're going to give this region a name now. We're going to call this the embryonic pole. It's like the North Pole, South Pole. We're going to call this the embryonic pole. This one we're going to call the AB embryonic pole or abembryonic pole, just like the South Pole, just so we can talk about different mutations and things like that. So the inner cell mass actually lives at the embryonic pole. Okay, so here's a cartoon of that. Now I colored these in. So there's our inner cell mass. There's our embryonic pole. Our AB embryonic pole will be down here. There's our blastocyst cavity or blastocele. We're all good, right? All right. And here's everything I just said about the poles. Inner cell mass pushes up as I just explained. And we get our embryonic pole up here to the North Pole. Our AB embryonic, abembryonic pole down there. 180 degrees away, just like the North and South Poles. Okay, there's another beautiful picture of a human blastocyst. Again, there's the inner cell mass, and I think you get the message. Now, the next thing that happens are we still have a zona pellucida right here. See that? Strong, looks pretty strong, doesn't it? It is pretty strong. It's followed us all the way around. So with the zona pellucida here, we cannot attach to the uterine lining. We need to attach to those epithelial cells on the uterus in order to start the process of having this baby continue. So in order to do that, we need to get rid of this darn zona pellucida, and that's called hatching. So the blastocyst literally hatches from the zona pellucida. About day 5 hatching occurs, maybe a little bit later in day 5. There's some enzymes that are released that bore a hole in the zona pellucida, and we'll see a real picture of a human embryo hatching here in a second. These enzymes in the hatching usually occurs over the inner cell mass at the embryonic pole. Look at this beautiful picture. So there's the blastomere, and there is our blastocyst literally squeezing out of a hole. It's released enzymes, it's bored a hole in the zona pellucida, and now it's squirting out. So that's a hatching blastocyst. Okay, and the enzymes dissolve the zona pellucida. I think everything I said. Okay, we're ready for implantation. What happened? We already said this slides out of place, right? Day 2, you lose the corona radiata. There's the corona radiata. Day 5, you lose the zona pellucida here. So we have a naked blastocyst swimming around in the salt water. Now, before we implant, a second round of segregation occurs. And this time it doesn't affect the outer periphery cells, the trophoblast. It affects the inner cell mass. We are going to split that inner cell mass into one group of cells that hug the trophoblast layer and toward the embryonic pole, and a group underneath that which are more toward the embryonic pole. So the top group closer to the embryonic pole, that's called the epiblast, and the group right underneath that is called the hypoblast. Epiblasts are more columnar type cells. They are more dorsally aligned because they're toward the embryonic pole. I'll show you a picture in a minute that will help. Hypoblasts are deep to that. Okay, now how do they do that? How do they segregate? Well, we got some more proteins that are going to be made by the central dogma. So the dorsal cells of the inner cell mass, they are going to secrete, in addition, they're already secreting nanog, but they secrete something called fibroblast growth factor 4, or FGF4. And FGF4 and nanog make them become the epiblast cells. Okay, now what about below the epiblast cells? Or how does it do that? So the FGF4 from the epiblast cells, they diffuse downward and they diffuse out into the interstitium. And they contact cells neighboring them, and if the FGF4 gets into neighboring cells, it causes them to secrete GATA6. Not themselves, though. The FGF4 is released outside of the cell, and it floats around, and if it contacts another cell of the inner cell mass, that one starts secreting GATA6. The ones that secrete GATA6 are going to become the hypoblast cells. The ones that just secrete FGF4 and nanog, they become the epiblast cells. Okay, got it? So to review, hypoblast cells will have to secrete GATA6. Epiblast cells have to secrete nanog and FGF4. Got it? Let's see a picture. So here is a inner cell mass, and it's undergoing segregation. The cells haven't lined up in their layers, but the nanog and the FGF4 secreting cells are the blue ones. These are going to be lined up right up here for the epiblast layer, and they're secreting that FGF4 and it's soaking into some of these cells and they're turning yellow. That's because they're expressing GATA6, and that's going to make them pull down to this region right here. So with a few hours go by, and now, did I make those colors? I did make those colors wrong, didn't I? Hypoblast should be yellow. I should have made that yellow down here, but sorry about that. But the GATA6 secreting cells form the hypoblast, and the epiblast is formed by FGF6 secreting and non-ONC secreting. Got it? This is a really important concept here, because now we have a inner cell mass that is two-layered. We have our epiblast and we have our hypoblast. This layer, these are real pluripotent stem cells. You can harvest these cells right here and you can make any tissue in the body. A pancreas cell that secretes insulin, a heart cell that beats, a brain cell. These are amazing cells. In fact, the entire body is made. These are the father of every cell in the body. It's going to change when this becomes a trilayer disc. This is going to turn into an ectoderm. But it's the same. There's different genes turned on, but this is a very important layer. Okay, everybody good with that? Okay, these are going to become what are called the germ layers. Okay, so we've just created a bilaminar disc. Bi means two, so we got a two-layer disc here. And yeah, sometimes it's called a bilaminar embryonic disc or bilaminar blastoderm or just a bilaminar disc. So that's an important concept. And we haven't even implanted yet, right? So around the time of implantation, now we actually have an up and down, right? If this is the embryonic pole, this is going to be the dorsal surface of the embryo. Down here is going to be the ventral surface of the embryo. In a minute, we're going to be able to show you which way is toward the head. We're going to form some AVE cells. Okay, but for right now, dorsal, ventral. So it's said that we have a dorsal ventral axis is now established, and we haven't even implanted yet. Okay, and now let's establish the cranial caudal axis. And to do that, we have to have some more cells created. So one more segregation story, and that's of the anterior visceral endoderm. So a third cell population, as these epiblasts and hypoblasts are kind of getting into their final positions, a third cell type emerges. And it emerges from the hypoblasts. Specifically, the hypoblast cells that are toward what's going to become the head of the embryo. They undergo a change. They undergo a transformation because some different genes get turned on, as we'll see here. And these cells, these anterior, and let me explain, let's stop right there. This is strange. So cranial, in anatomy, cranial means up, superior, right? In embryology, cranial is an AKA for anterior. In anatomy, cranial is up, superior is up. Frontwards, like the front of your body, that's the anterior portion. Posterior is behind, inferior or caudal is down to the floor. But it drives you crazy when you try to read these books because cranial and anterior are AKA in embryology. And I think that's because the embryo is going to fold and this cranial is going to eventually become anterior. So watch out for that if you're reading this stuff on your own. Cranial and anterior are AKAs. So this third layer of cell comes from hypoblasts. Specifically, it comes from the ones that are located at the cranial end of the embryo. Okay, they undergo a transformation and become these AVE cells, or anterior visceral endoderm cells. Okay, what causes the transformation? Remember, they're secreting nodal. You're going to block nodal, and that's what we're going to do. So to make an AVE cell, nodal must be turned off. Nodal, by the way, is a TGF beta, transforming growth factor beta family. It's a very important, we'll talk about this a lot as we go through this course I'm putting together. So you got to turn that off. So, okay, we'll turn it off. It's not so easy. That's a powerful gene. It's hard to turn that off. So we got to have a couple backup systems. But the first thing that happens is these cells in the anterior part. Do I have a picture of those yet? Where are they? Yeah, they're going to be right here. So those cells, they are going to secrete a protein called DICOF1 or DKK1. Now that doesn't directly turn off nodal, but it does block the expression of the molecule needed to make nodal. WNT or WNT is needed to make nodal. We didn't talk about that before, but we'll talk about it now. So DICOF1 blocks WNT. If you block WNT, you can't make nodal. If you don't make nodal, then your hypoblast cells, they take on different characteristics, and we call them the anterior visceral endoderm cells. Okay, play the tape back. I know that's kind of tough. So, and then we're not even done. So I said there's backup systems. Here's the backup system. So other genes also turn off, are also turned on in AV cells. They also help block the expression of nodal. Nodal is tough to turn off. So in addition to DKK1, you have lefty 1 and cerebrus 1. They're both turned on, and all three of them, DKK1, lefty 1, cerebrus 1, all of them finally get nodal shut off, and those cells take on a different morphology, and they become AVE cells. Good? So this is what we've built so far. This is our blastocyst, and yeah, this is our blastocyst. So we have a blastocyst cavity here. We have a trophoblast layer around the outside. The inner cell mass is no more. It's split into two groups of cells, epiblast cells, hypoblast cells, and the hypoblast layer has now morphed into the anterior visceral endoderm cells. So that's what we built so far. Let's keep going. And this AVE area, what does it do? It's really super important, and we'll talk about it more when we get to the primitive streak. But it single-handedly stops the formation of the primitive streak in the cranial end of the embryo. You don't want it forming. The primitive streak forms in the caudal end. And why doesn't it form in the cranial end? Because of these AVE cells. They stop it from doing that. Okay, the AVE cells also differentiate into the cranial-most portion of the AVE cells. Those actually go on to help make the heart. It's a signaling center. There's all kinds of proteins and transcription factors being turned on there, and it's very important in the formation of the heart. The more posterior, the more caudal end of the AVE cells, they go on to help form what's called the precordial plate, which we'll talk about in the next video. Precordial plate is super important. That's going to form part of your brain. It forms the forebrain and part of your head. So that's what those AVE cells do. All right, binding. So finally, we've got quite a complex little zygote already, don't we? A lot of gene transcription going on and a lot of different cell types, and we haven't even bound yet to the darn endometrium. But let's do that and get on with this. So before we do, the uterus is being prepared for all this, mainly by progesterone and estrogen, which are being released from the corpus luteum of the ovary, and you'll get that in a different course. I'm not going to dig into that. But those will prepare. And one thing they do, and the presence, we don't quite know how this works, but after the olive drops in the martini glass, that stimulates the uterine lining to stop making a slippery substance. It's like WD-40 here, this mucin 1. And the combination of them stop mucin 1. And mucin 1, if that was being secreted by the cells of the uterus, you couldn't implant, you'd slip right off. So if you turn off mucin 1, now you got a good chance of sticking and binding to the epidermis layer of the endometrium. So mucin 1, it's a glycoprotein, normally protects the uterine lining and makes it slippery. But the binding takes it down. The cells don't secrete it anymore. So let's officially bind. Now we're about day 6 or day 7. It's time to bind. And remember, hatching has occurred. We are out. The zona pellucida is behind us, and our blastocyst is ready to bind. Now it just doesn't bind. There's some receptors that does the binding. So the big one on the trophoblast is called L-selectins. So these are the binding receptors on the trophoblast, or on the zygote, if you will, or on the embryo, LAKAs. So those are going to hook up with receptors on the epithelial cells of the endometrium. And those are called trophinins. And there's also some other ones we won't get into on the trophoblast as well. But trophinins and L-selectins bind together. Let's see a picture of that. So there's a L-selectin. Here's our blastocyst, and it's binding to a trophinin right here. And great. Now this thing is going to be sucked down inside the endometrium here in a second. That's the next job. It's got to get inside the endometrium. Okay, so official binding between the blastocyst and the uterine lining. Good. Let's take a sidebar here. Ectopic pregnancy. Normally, by the way, this binding right here, normally this occurs on the posterior wall of the body of the uterus. Sometimes it doesn't. And sometimes it's a problem, sometimes it's not a problem. It depends where it binds. If this binding right here, if this occurs in the fallopian tubes, it's an ectopic pregnancy. In fact, an ectopic pregnancy means that you have not bound to the posterior wall of the uterus where you're supposed to. That can be very dangerous. Remember, we have cell division going on here. This thing is going to start growing like crazy as thousands of cells develop. And if this happens inside that little tube, you can blow that tube right up and cause horrible pain and bleeding and all sorts of things. So let's go through this slide. So if ectopic pregnancy means that the binding can occur in the fallopian tubes, the peritoneal cavity, it can attach to intestine, it can attach to the ovary. I'll show you all the places, but these are all examples of ectopic pregnancy. If the zygote does not bind to the lining of the endometrium, you have yourself an ectopic pregnancy, and it can kill the mom and the baby. So this is from Carlson, liking this book more and more. The most common site for ectopic pregnancy is the same place fertilization occurs. Remember, fertilization occurs in the ampulla. About half of the ectopic pregnancies, the darn fertilized zygote didn't make it all the way down and go kerplunk in the uterus. Instead, it attached to the ampulla of the fallopian tube and is growing here. It burrows into the fallopian tube and starts expanding, and very soon the mother has horrible abdominal pain and starts bleeding from this. You can't have a baby here. 25% of the time it's more at the isthmus of the fallopian tube. It can occur in the cervical region here, which is not a great thing. I think it's called placenta preva can occur, where you break the placenta starts herniated out of the os in the cervical canal, external os, internal cervical canal through here. So that's a dangerous one. And here the egg didn't go in the fallopian tube. The Fembriae missed it, and the egg attached here, and the sperm homed in on it, and they bound to it here, fertilized it here, and you have a baby growing here. There have been babies, they have to be taken up by C-section, of course, but there have been successful pregnancies here. Not many of them. The most common type of this ectopic pregnancy that's not even in any of the normal plumbing is one that occurs in the pouch of Douglas. Remember, we learned in Gross 2 anatomy, this is one of the lowest regions and can collect with blood and pus and things like that. Gravity helps the egg if it doesn't get, or if the oocyte doesn't get into the fallopian tube like it's supposed to, it gets put down in here. The sperm can come right out the tube and find it. So about 1% of ectopic pregnancies occur here. Okay, you'll get more of that. in gynecology, but that's not till I think about ninth quarter, seventh quarter, long ways away. Okay, so we're bound to the endothelium of the endometrium. And now the job is to penetrate. We have to get into the stroma. That's like the basal lamina that you know from histology, that epithelial cells sit on a basal lamina. It's called a stroma, and it's pretty deep in the uterus. All right, so the implantation stimulates the trophoblast at the embryonic pole to grow a new layer of cells. We got to pull that embryo inside the endometrium. To do that, we need to grow some new cells. And the trophoblasts, they literally go, they're like stem cells, right? All these things are like stem cells. They literally start making a new weird cell type. And this is called the syncytiotrophoblast. Almost like a cancer, I want to say. It's crazy. Let's talk about it. A very weird tissue. It's very aggressive. It really burrows itself into the endometrium, and it pulls the zygote with it as it burrows. So these are weird because they have nuclei, but they lose all their cell membrane. So it's a bunch of nuclei floating around in this weird tissue. So there's not a lot of individual cells. So it's just a very strange and fast-growing tissue. It has some leading tips. Those are going to push their way between the epithelial cells and get into the stroma and start digging. They secrete a very powerful proteolytic enzyme called metalloproteinases, very destructive, and they rip apart the cell adhesion molecules between the epithelial cells, and it pulls that zygote inside the endometrium. It also, the contact, and we don't even know what causes this, but it also causes the death of some of the endometrial cells as well. So it can help the zygote pull itself in. So it's a crazy tissue. Here's the very birth of it. So here's the cavity, the uterine cavity. We've already abound. And here's the trophoblast layer, and it's secreted this weird new tissue. You can see the nuclei, but the cell membranes are gone. That's the syncytiotrophoblast, and it has these points where it pushes between the epithelial cells, and this is going to get so big and get growing so big, it's going to surround this whole thing eventually as we go on. So everything we said already, the invader, it pushes its way through the epithelium of endometrium, secretes MMPs, and it stimulates some of these cells. It just kills some of these cells, just die. It puts them into apoptotic mode, and they die, and we're not exactly sure how that mechanism works. Now look, maybe a day later, now look how far it's in there. Here's the uterine cavities out here. It's the syncytiotrophoblast has gotten huge, right? It's even starting to develop these little lacunae, which is going to become the first blood supply here. There's the mother's, the circulation. There's the arteries, and there's capillaries we can't see, but that's mom's blood. We need to hook those up too, right? Because this tissue is growing. It needs to have blood and oxygen and have its waste removed. So we're going to hook that up in a little while, but it's pulled itself in there. Okay, side note here, we got to talk about the pregnancy test. The pregnancy test is because of this tissue right here. This tissue secretes human chorionic gonadotropin, HCG. And there are tests you can buy right over the counter that can detect the HCG in your bloodstream. And it can tell you with pretty good accuracy these days if you're pregnant. Where does it come from? HCG again, it's from this crazy syncytiotrophoblast cells. So they do some other things too. The HCG, it's a hormone, so it circulates through your body, through the bloodstream. It finds the corpus luteum in the ovary, which is secreting progesterone and estrogen. Normally, that corpus luteum doesn't stay alive very long. But if the syncytiotrophoblast secretes HCG, it reaches the corpus luteum and gives it supernatural power, and it doesn't die off. It stays alive for a long time and helps support the pregnancy. Okay, so eventually the placenta is going to secrete pregnancy hormones and progesterone, or is going to secrete progesterone and estrogen about 12 weeks away from that. So it turns the corpus luteum, it stops it from dying and makes it superhuman. So HCG, a pregnancy test, and it keeps the corpus luteum alive longer than it's supposed to be. Now we got some house cleaning to do. A simple name change has occurred, and I go on a rant here. The authors are absolutely terrible. You ever tried to read an embryology book, it'll drive you absolutely crazy because of this term, cytotrophoblast. But once the trophoblasts, or once the syncytiotrophoblaст cells have occurred, have grown, and remember the the trophoblast cells are the ones that actually are giving rise to this stuff. These cells are splitting and being pushed out here. So once the syncytiotrophoblast is here, you technically shouldn't call it trophoblast layer anymore. They changed the name to cytotrophoblast. But it's really the same thing. And again, authors are terrible. They'll flip, even in the same paragraph, they'll call it trophoblast, and a few words later, they'll call it cytotrophoblast. Drives you crazy. But they're AKAs for each other. Okay, so there's some other stuff. Now, let's go back to our undergrad immunology, or even biology. This has got, like the cells here, this has got dad's DNA in it, right? So as this starts to hook up to the bloodstream, the body's immune system is going to take a look at this and say, hey, wait a minute. This is not self. This is not part of mom. We don't know who this guy is. Let's attack and kill this thing. And so we need to fix that, right? Otherwise you won't get pregnant if you mount an immune attack against this. And inflammation can destroy it. So we need to shut off mom's immune system right here. And that's done by a couple of things. The embryo, we're not exactly sure where this comes from, but the embryo, some of the cells in it secrete something called early pregnancy factor. And this is thought to act as a very localized immunosuppressant, and if an inflammation attack occurs, it calls that off. So that's one. The more important one, though, is released from the stroma of the endometrium itself, and something called interleukin-2 is released. It actually comes from white blood cells that get in the area. Leukocytes move into the area. I think we'll cover that one more time. But interleukin-2 gives the zygote a pass. It turns off the inflammatory response. So it protects your zygote or your embryo. It protects it from being destroyed by your mom's immune system. So that's an important thing. Okay, time for how about a new cavity? We talked about the blastocyst cavity. We haven't talked about that for a while. Let's make a new cavity. So about day 8 now, and now our embryo is well implanted by now, but we're going to form a new cavity, and this time it's going to be between the cytotrophoblast. Remember, we just changed that name, right? It was just trophoblast, but I'm going to try to stay correct. Cytotrophoblast and the epiblast layer, a new cavity. This cavity is going to be the amniotic cavity. Guess what kind of fluid it has in it? Amniotic fluid. That kind of sounds familiar, doesn't it, from somewhere? So let's take a look at it. So now our blastocyst is almost completely implanted, right? It's still got a little ways to go. Syncytiotrophoblast is going crazy. Here's a maternal capillary. It's going to punch a hole into that pretty soon and get some blood from it. But now we got a little cavity forming right there. And that is the start of the amniotic cavity. Okay, amniotic cavity. That'll eventually grow huge and encompass the entire embryo. We'll talk about that as time goes on. It's not very well studied in humans, so they're not exactly sure. Research on primates kind of tells us a story. But as that amniotic cavity forms, the trophoblast cells, those cells right on the top here, these, well, I guess we should call them cytotrophoblast cells. Um, or no, is it from, let's see, I even get confused on this myself. Better stay on my slides here. Almost immediately, the amniotic cavity gets lined with cells. They migrate from the epiblast. So these cells are coming from the new epiblast cells themselves. They're splitting through mitosis, but different genes are turned on, and they're going to go up and form a lining around that. And that's just called the amnion. It's a lining around the amniotic cavity. Okay, the cells are called amnion cells that make up the amnion. There's some AKAs. Sometimes they're called amnioblasts. Uh, amniogenic cells are some kind of odd. These are not mainstream books, but these authors use these terms. But amnion cells make up the amniotic cavity. Okay, so they make up the amnion. The amnion is important because that is one of the 4 extraembryonic tissues or membranes, and it's considered, Carlson considered it an extraembryonic ectoderm, and we'll kind of talk about that in the future. So here's our amniotic cavity. This is kind of a child's drawing. Here's our hypoblast. Here's our epiblast layer, and then we formed a cavity in between the cytotrophoblast layer, remember, and the epiblast layer. We formed a cavity, and then these cells, through mitosis, made new cells which formed a nice lining here, and those are the amnion cells. Okay, we good? And we're still not fully implanted. The uterus cavity is out here. The martini glass is out here, so we're moving into the endometrium, into the stroma, but we're not quite there yet. Further invasion, so let's get there. Let's pull ourselves all the way into the endometrium. So the syncytiotrophoblasts are going to increase in number and volume, and as they get stronger, they literally pull that embryo inside the endometrial tissue, or they invade the endometrial tissue like the army of the dead invading the land south of the wall, right? That's the Game of Thrones. Did you guys like the final episodes on Game of Thrones? Thumbs down for me. I was not happy with it. Anyway, by day 12, the embryo is completely embedded in the endometrium. So week 1 has went by, so it can eventually be pulled all the way in. So here's by day 12. Here's the cavity, the endometrial cavity out here. This is where it went through. There's the epithelial cells of the endometrium. We're in the stroma layer of the endometrium now. And it's complete. Look at the cytotrophoblast. Went wild, hasn't it? All this purple completely surrounds it now. And we got these lacunae. We'll talk about those next. And but yeah, completely embedded. Hooking up the blood supply. So let's talk about that. We kind of talked a tiny bit about it. But as that syncytiotrophoblast develops, we start getting these cavities. I mentioned these are called lacunae, or more specifically, trophoblast lacunae. So they're empty right now, but they're not going to be pretty soon, because this invading syncytiotrophoblast is going to poke a hole in the endometrial capillary here, and once it pokes a hole in there, this is going to fill with blood. So this will be our first makeshift circulation of our embryo. Okay, so these projections of syncytiotrophoblasts erode the maternal capillaries, and they're going to hook up our system here in a second. And here's a nice picture of them actually. Syncytiotrophoblast, the purple, has gotten into the mom's capillary, whether it be a venule or an artery or capillary. It's bonded to it, and now we have blood flowing into those lacunae. And so, yeah, that's our first blood system. So now we have blood in here. The blood, oxygen, for example, can start diffusing in here all the way into here. It can feed the bilaminar disc. It can feed all these cells. If we have waste products, that can diffuse back through here and get into this and get it through the venous system, which we can't see. So we've just created our first blood supply. That's called the Uteroplacental circulation, which was just born. So everything I just said, the endometrial capillaries are now feeding the lacunae. So mom's blood is starting to flow. Everything I said already. Okay, now you got to know what the decidual reaction is. So as this blood circulation is going on, the stroma of the endometrium is also changing because of implantations. A lot of the cells that make up this stroma are fibroblast-like. They're starting to feed and fill up with glycogen and fat. They're almost like little livers. That's where we get glycogen from. So they're little storehouses, and we could feed the cells because our growing embryo needs to have sugar, right? And you can break down glycogen and make sugar from that. You can break down fat and make sugar. So that's where that comes from. Those are no longer called stromal cells. They're called decidual cells at this point. And they can be used to feed the cells of the growing embryo. Okay, everything we said, I think, they used to be the fibroblast-like cells, the endometrium. The presence of embryo plus progesterone converts them into these secretory cells, which you can break down and take the lipid out of there and the glycogen. Kind of everything I already said. They do one more thing though. They do secrete. They're thought to secrete some growth factors. The metabolites are probably breakdown products from the fat and the glucose, or in the glycogen. But there are some growth factors, so it does help support growth of the growing embryo as well. Those are decidual cells. They used to be just regular old stromal cells of the stroma. Okay, and important. So as this decidual reaction is going on, as these cells are swelling up, something else happens. So leukocytes now come into the area as part of this decidual reaction, and these leukocytes secrete interleukin 2. That also prevents the mom's immune system from attacking the embryo, right? Let me jump back here real quick. Yep, that's the same one that we talked about right back here. Just wonder, I thought I might have said interleukin 1, but I didn't. Sorry about that. We're good. All right, so part of the decidual, we're almost done. Part of the decidual reaction then is these weird leukocytes coming into the area, secreting interleukin 2, and that's telling the mom's immune system, hey, back off, leave our zygote alone, don't kill our embryo here. You know, this is a new life, and that's super important. This is supposed to be the most important function of the decidual reaction, according to Carlson. It's called granting immunologic privilege to the zygote, and super important. Okay, we'll see you in the next video.